I'm very happy to have Abby here. So she's an associate professor in Albion College. And so Abigail and I, we met when I was still a PhD student in Marseille, France. And she was a postdoc already there. And so at the time, I guess we were both working on similar things. We were both doing population genetics and metabarcoding at the same time. And we were very interested in patterns of biodiversity, both at the intraspecific level and at that interspecific level, trying to understand how those potential correlation between those two levels can be generated and what are the process behind those. And so Abby had also another project that is still ongoing, and she's going to talk about it today, which is about cryptic species, and in particular in the marine world. So yeah, Abigail, the floor is yours.
So thank you for having me. Thank you for coming today. I'm really excited to see so many people here. And I'm excited to be here. Before I went to Marseille, I did my PhD at Stony Brook University and heard a lot about Dauphin Island and the folks here through the people there who had connections. And so I'm excited to finally be here and get to see the facilities, walk around a little bit and meet with you. So like Aurelian said, this is a project that started a long time ago. We started this 10 years ago. Part of it finally came out about 18 months ago, but this is the project that keeps growing. Every time we look at the data differently, or I talk to people about the data, we come up with new ideas. What we are doing here is trying to understand patterns of cryptic species in the ocean. On a broad scale. So I'm not going to be showing you a particular case study. I'm going to be talking at a big level. And if I have time, I'm keeping an eye on the time. If I have time, I will hopefully get into some of our thoughts about processes of formation here as well. But I'm going to start with patterns. And I need to start by actually defining cryptic species. And what we're talking about here are species that can't easily be classified based on morphology, but which are genetically distinct from each other once you actually sort of look at what is going on under the surface. There's two of my favorite case studies up here from the mollusks. We've got two species of Glaucus nudibranch on the left. To me, those look very, very different, but they were originally described as the same nominal species. On the right, we have two species of Crypigula, both originally described as Crypigula plana. And it turns out that these have geographically distinct distributions. They have some morphological differences, and really importantly, reproductive differences as well. So they have now been formally given different names. So that's what we're talking about here today. The first big review of marine cryptic species is now over 30 years old. Nancy Knowlton in 1993 published this paper, Sibling Species in the Sea. And yeah, so where are we over 30 years later, given the increase since the early 90s of molecular data, which allows us to find more and more cryptic species? This figure is coming from a review that also is getting up there. This is a 2018 review looking at just the incidence of the phrase cryptic species in the literature. It's not the only thing they did in the paper, but that's the graph you're looking at. And so you can see that we have basically cryptic species increasing rapidly through the early 2000s up to 2015. The light green line is, I think, a slightly different associated search term, but I don't remember what it is off the top of my head. But the point here is that we have this rapid increase. This is consistent with the rise in molecular data, and I think we know that makes sense, and also has continued increasing to today. Why does this matter? So we have this sort of historical context. Why do we care about this? I'm first going to show you a little toy example. I want you to imagine going out in the field and measuring some ecological parameters. You might look at this picture and count that there are two different species here, and estimate our abundance of triangles at 4. But if it turns out that the triangles are actually two different cryptic species, we have misestimated both of those parameters. What we've done is underestimate species richness and overestimate the number of triangles. that are out there. And so this is just a toy example, but there are examples of this happening out in the field, and there's no reason to think that this doesn't frequently happen out in the field. Here's some real data coming from a paper looking at coral reefs and genetic distance on coral reefs as measured with FST. And what this paper did, I was very happy to see this come out. What this paper did was to actually calculate FST values on patches of reef if you ignore cryptic species and just assume you're all dealing with the same sort of morphotype as a species. You get relatively high values of FST, indicating low levels of genetic connectivity. But it turns out what that's actually measuring is difference between species. And if you correct and say, in fact, we do know we have 2 cryptic species here, you can see that those estimates in this case actually overestimate the genetic differentiation between populations. So we know that there's examples out in the real world where this matters. With 30 years having passed, more than 30 years having passed since Nancy Knowlton's review paper, we can start to ask questions about patterns in marine metazoan cryptic species and what generalizations can we start to make about this. So we now, in 2020, is when really the literature review was conducted through, was 2020. We have lots and lots and lots of cases, now enough to make some generalizations. And so what I'm presenting in this part of the talk is work that was published, came out early last year in Biological Reviews. So if you want more details, particularly about the methods, they're in there. I'm going to walk through some stuff. We are talking about marine species, we are talking about marine animal species, and we eliminated parasites from our data set. We're really talking about free living species, partly to make our literature search easier, and partly because we anticipate that one parasitic species might be more likely to be cryptic for a lot of reasons, but might also have some cryptic diversity in response to, say, cryptic diversity in their hosts. That was just one more level of complication than we wanted to deal with. So we're dealing with free living marine animals here. And I need to acknowledge my co-authors here on the talk. So I've got several hypotheses. This is going to be kind of a wordy slide that I'll walk you through. What are we testing and why? The first hypothesis is maybe sort of self-evident. The presence of molecular data, more or less by definition, makes it easier to find cryptic species. And genetic data are often used, usually used in cryptic species delineation. So that leads us to the prediction that species with molecular data are going to be more likely to contain cryptic species within a nominal species. Another thing related to this molecular piece is maybe there's other technological constraints leading to cryptic species. One thing I often come back to is that microscopes have improved since the 19th century. And so maybe species that were described earlier are going to be more likely to contain cryptic species just because our ability to look at things has improved. To me, as a biologist, this maybe is the most interesting. Maybe there's actual biology underlying the presence of cryptic species. Might some traits be easier for humans to use in species delimitation? These traits might not correspond to what organisms actually use. And so we thought about this a little bit and thought about how taxa are described in particular clades versus what we know about how the organisms actually function, and came up with some hypotheses. We predict that we might find fewer cryptic species in groups with hard skeletons because taxonomists might have an easier time measuring things or just, preserving things. Taxa with internal fertilization. If we think of genitalia as being important to call species differences, and we know they're used in many taxa for that, maybe internal fertilization makes you less likely to contain cryptic species. Same rationale for external genitalia. If we're using those genitalia for species delimitation, species or groups without them may be more likely to contain cryptic species. And this thing about image forming vision, if we think that vision might be important in preventing hybridization in the field, maybe having better vision makes you less likely to have cryptic species. We also thought, well, okay, again, with this history of science, maybe the prevailing ideas about evolution at a time when species were described affects whether or not these taxa are later found to contain cryptic species. So another way to put this is that changing species concepts might lead to scientists lumping taxa or splitting taxa. We know that you can think about your taxon of choice and sort of the waves of lumping and splitting that may exist through time. One concrete prediction we could make out of that is that maybe species described before the Origin of Species was published would be more likely to contain cryptic species. We also lumped in here expect that species containing cryptic species might have larger ranges. Again, if things are described earlier or in a period of time when allopatry isn't to be very important in driving speciation, stuff might get lumped together across a broad geographic range. So I'll come back to each of these predictions. I don't expect you to remember them all, but just to say we set up these predictions with a mix of sort of history of science, human components to this, and maybe there's some biology underlying it as well. How did we start to test these? Well, we assembled a giant data set of data sets. So let me break down this methods into 3 different slides. The first thing we did was a systematic review of the literature to identify cryptic species. We first came up with some keyword searches in Web of Science, and then we also went and looked at all of the papers citing either that 1993 paper by Nancy Knowlton or a year 2000 paper on the same theme that's also often cited. We combined those, looked for and removed duplicates, and did all of the sort of systematic review steps, screened abstracts, read full text. At the end, we wound up with 977 distinct cryptic species complexes. So another way to think about this is 977 nominal species that have been split into at least 2 and sometimes more than 2 cryptic species when you look more closely. So that's our cryptic species database. 977 nominal species. Actually, before I get there, this is cool. I presented this. We started to write this up. I was really excited about the results we found when we analyzed those data. And very quickly, it became clear that we need something to compare it against. So we have these 977 cryptic species, but so what if we don't know sort of more about what's out there in the ocean? And that brings me here to leveraging a lot of other publicly available databases to make some comparisons. So we're looking at now all of the nominal species in the World Register of Marine Species database. For this, we asked some questions for each of these species. For each of these species, is there genetic data in NCBI? So for instance, you can see we have over 187,000 species in worms. 38,000 of them have records in NCBI. A relatively small number, about 50%, have data in the Ocean Biogeographic Information Systems, or OBIS, so that's geographic distribution data. And we also collected some other information out of here, like year of description, genus size, some other things that we can just get from the worms database. The biology is a little bit more complicated. There is no reasonable, reliable database that gets us species level identification on the whole worms database. So to think about biology, we grouped by taxonomic class and considered traits that by and large are fixed within a class. And so for each class, we wanted to know about those traits that I mentioned already. Do these things have a hard skeleton? What about their reproduction? What about their vision? So if I think about this and summarize all of that, for each free-living marine metazoan species in worms, we know some stuff. We know whether or not our literature review identified cryptic species. We know the year of description of the taxon. We know whether or not there's genetic data present in NCBI. We know some geographic information for about half of the species. That's actually the one for which we have the most missing data. We've got some biological trait data. And we also were able to, for 57 of the classes, come up with an estimate of a dated phylogeny using the program time tree. So we have all of this information. What we then did with it is a whole bunch of logistic regressions and using Bayesian information criteria to figure out the best model. And the best model for what? Well, we are trying to explain the probability of finding cryptic species in a nominal taxon. So given all of the information on the previous slide, what is the probability that we find cryptic species within a nominal species? So what did we find? First thing, least surprising thing, nominal species with data in NCBI are more likely to contain cryptic species than those without data in NCBI. Again, exactly what we expected, but we did need to check. And we can put some numbers on this. So here are all of our species in worms and the percent containing cryptic species. If a nominal species has data in NCBI, we see about a 4.5-fold increase. That probability of containing cryptic species goes up about 4.5 times. And if the species has both nuclear and mitochondrial data in NCBI rather than just one or the other, we're looking at about a 6-fold increase. Okay? But if you're looking at the y-axis, you can see that overall we're dealing with relatively small percentages of species in worms that do actually contain cryptic species. And I may come back to that point later. This was the strongest factor in the models, and again, maybe the least surprising of all of our results, but we did need to think about it and sort of include it in our processes. We also found evidence that the history of science is important in various ways, and the way I'm highlighting here is that taxa, again, nominal taxa containing cryptic species tend to be described earlier. You're looking here on the screen. I will use the mouse maybe for the folks on Zoom. We've got light gray bars here. Those are taxa that contain cryptic species, on average described earlier than taxa that don't, nominal taxa that don't contain cryptic species. So that fits along with our predictions. We found that nominal taxa containing cryptic species have larger ranges. That's true. Again, light gray is yes, cryptic species, dark gray is no. That's true if we just look at latitude. What's the latitudinal range? Just kind of a brute force, most northern, most southern. It's true if we look at the longitudinal range in the middle panel. And if we take those two ranges and put them together into a very crude estimation of a convex hull, it is still true. So all of these ways to think about range, if you contain cryptic species, your range is larger. This also kind of makes sense. So that was exciting. These factors in our models were all stronger than the biology, but we did still find signs that biology matters, which again was exciting to me. Harder skeletons, this matches our prediction. Groups with hard skeletons, less likely to contain cryptic species. Groups with image-forming vision, less likely to contain cryptic species. Fertilization, if you are an external fertilizer, or there's a couple of classes where fertilization mode really is variable, so variable that we scored it as both. Those also have more cryptic species. And here I'm thinking about polychaetes and gastropods off the top of my head, tend to have more cryptic species. Here's our tree looking at this, and I want to just highlight a few things. Bony fish, fewer cryptic species than expected. Based on all of the information that went into our models. And that was the group that was the lowest, that was the low outlier. But our malacostracans, including our decapod crustaceans, also had fewer than expected. On the high end of things, hydrozoans is our highest, like more than expectations group, followed by polychaetes and other sort of squishy stuff. Scyphozoans are here. Ophiuroids are not squishy, but they're still cool. All of these things that I just put up have more cryptic species than expected based on all these other variables. So to bring it back to the hypotheses for this first sort of large chunk of the talk, again, here's our first prediction. Genetic data, presence of genetic data makes it easier to identify cryptic species. Yes, absolutely, very clear. Species that are described earlier do have more cryptic species. Things are a little bit more, a little bit sort of trickier in the biology. We do find support for this hard skeleton and image forming vision. We did not find any sign of genitalia in the models. And I have a little yellow line here next to the internal fertilization just because of that class. If you separate the organisms into internal, external, and both, we don't see the effect. You have to actually lump external and both types of fertilization together. We did also, I didn't really show you about the species concept. Ask me questions or go read the paper, but we did very clearly see a signal that species, nominal species containing cryptic species have larger ranges. So in general, our predictions are supported. And we kind of summarize this in the paper as saying, well, we do have strong support for the role of scientific history, the role of humans actually making these species determinations in the presence of cryptic species within a nominal species. But we see the beginning of support for the idea that biology may actually play a role once you take into account all of these other sort of human caused errors and biases. Okay, so that's the sort of first chunk. And having gone through it, we now have this database of 977 nominal species containing cryptic species that we can start to do some exciting stuff with. And I'm going to present definitely one and maybe two things we are doing with this big data set now that it's there. And the first question we've been asking, okay, we have these results, and maybe what you're sitting out there thinking is, okay, but I study X taxon. What does that mean for worms or for oysters or for, you know, whatever cool things you're studying? Where are we most likely to be missing cryptic species and therefore potentially making errors of the type I started with? So I'm going to address that starting here with the presence of NCBI data. We know that the presence of NCBI data really strongly influences whether or not we find a cryptic species. So I'm going to flip this around and turn it into a question. How many cryptic species would we have in a phylum or a class or a geographic area if there was genetic data for everyone? Because we know that there's not genetic data for everyone in NCBI. So to put this, I took this question and tried to turn it into something quantitative. I'll show you an example of the calculations from the mollusks, again, one of my favorite groups. Here's what we found in our literature review. So here's what we have so far. In our database of 977 species, 173 of those observed cryptic species are in the mollusks. If we look at all of the species in worms with NCBI data, we see that there's 7,904 species with NCBI data in there. So total pi is 7,904, 173 of those are cryptic. And if we make the admittedly big assumption that we can scale this up and say, okay, here's all of the species in worms, and we're making the assumption that that blue slice of the pie stays proportionally the same, so that the rate of discovery of cryptic species doesn't change, we come up with an expected value of 1,073. And so I can think about that and say, okay, that means we're expecting 1,073. We haven't found 900 of them yet. And I'm going to convert those numbers into percentages because, of course, these groups are very differently sized. So when I do that, we can say, okay, about 84% of potential cryptic species in the mollusks are still out there somewhere. So when I did that across different phyla, the one thing that popped out very strongly is that proportionally far fewer missing chordates down here. So we have proportion missing over here on the left. Far fewer missing chordate species than any of the inverts. Our bryozoans are up here. I think that's something like 94% missing when I crunched the numbers this way. And that doesn't surprise me too much. A lot of our fouling organisms are up there, are way up here. So I also broke this down into the class level because speaking of fouling organisms, the chordates, of course, are fish, and then we've got some invertebrate chordates. And so I split this into a class level analysis. So pick your favorite class. We see the same visible pattern at the class level. Species that are relatively low, we've got our bony fish, our elasmobranchs. Cephalopods are interesting. They're obviously invertebrates, but we can talk more about that later maybe. And then at the upper end of things, copepods now at the class level actually pop out as the class with the highest percentage of missing people, again, up in the 90s, or missing cryptic species up in the 90s. Here are our ascidians, our invertebrate chordates. So we have bony fish, we have ascidians, we have elasmobranchs, all of them kind of combining to make that chordate number. So that is exactly why I did the class level analysis. And sponges as well are popping out here, our demo sponges. So that's our sort of taxonomic we're still thinking about this and what this means, but a lot of it kind of intuitively makes sense. And I'm also, what I'm starting to do now, and this is really sort of hot off of the code is still broken stage data that you'll be seeing. We have more cryptic species that have been found in the Pacific, but we can think about this in terms of what's missing. So now I'm moving to geography. On the left, we have how many cryptic species did we find that are present in each of these oceans? And we use the OBIS data for that. More cryptic species identified in the Pacific. But again, Pacific is big. We know there's really high species diversity there. We somehow need to control this. And so if we look at ocean scale information and do that same kind of pie chart correction, we can see that in fact, where we are missing the most is up here in the Southern Ocean. So this is the largest proportion still missing in the Southern Ocean, right? And then fewer, interestingly to me, in the Arctic. And I think there's something going on here where a lot of the cryptic species that have been identified in the Arctic are fish, actually. And they're just thinking about sort of history of fisheries in the Arctic Ocean. We actually have more data here, but thinking about the Southern Ocean as a problem of access also kind of makes this make a lot of sense. We have not had as many sort of expeditions and sampling in the Southern Ocean as in other places. So where I'm going next, and this is where stuff starts to break down, is like that's cool, five ocean basins or whatever. But let's think about this on a finer geographic scale. And so right now I'm trying to, like, as we speak, trying to figure out how to do this using the 12 marine realms from Spalding's 2007 paper. And I will show you some data, but there are some major caveats with what I'm about to show you. First of all, our results are only based on about 5,000 animal species because my code keeps stopping because it's so big. And also, this is maybe a bigger caveat. I knew I had a couple regions that weren't working well in my code. And then when I looked at where they are unfortunately these areas. So I will show you data, but not from the temperate North Atlantic or from the Indo-Pacific. When I do that, again, we have very high proportions of missing species in the Antarctic and the Southern Ocean region, and lower proportions of missing species in some of these other areas. But having only gone through about 25% of the species I hope to, I don't want to say anything definitive about that. So, but I hope what this can summarize, in this part of the talk is to say that, okay, proportionally compared to the species diversity that's out there, cryptic species have been identified proportionally most often in chordates, especially fish, especially bony fish. And thinking geographically, most often in the Arctic Ocean and least in the Southern Ocean, which I think am currently thinking about as questions of access. So that's one way that we're moving. And now I do, in fact, have enough time to talk a little bit about process, which showed up in my title. So we know something now about patterns of cryptic species in marine metazoans. Where are they found? Where might you expect to find them? Where might you expect them to cause problems in your work? But I'm an evolutionary biologist. Remember, this work started in my postdoc when I was in Marseille. My postdoc advisor is an evolutionary biologist. And so we have been interested in process. Can we start to use these data to understand process of cryptic species formation and move beyond patterns? There's multiple processes that might lead to cryptic species formation, and people have argued about this in the literature. One is just straight up human error, right? And I talked a lot about that in the first part of the talk, that maybe just technology isn't good enough. People weren't looking at sequencing DNA in 1800s. So, maybe we just have process errors from humans. But it's also possible that biology actually matters here, right? So, and we might think that these eco-evolutionary processes wind up being important in a couple different ways. One, stabilizing selection might happen. And this is certainly a discussion in the literature. You might expect that stabilizing selection, for instance, in soft sediment habitats for burrowing organisms, might cause things to converge and stabilize in an optimal phenotype. Another thing is that we know a lot of population sizes in marine organisms, thinking, for instance, about those copepods again. We have very large population sizes often in marine organisms, and maybe large population sizes can prevent fixation or delay fixation of differences. And so thinking about this as a more neutral, which I put in quotes because I'm not necessarily thinking about neutral theory, but thinking about this as a non-selective process. Another thing that's out there is that, of course, these might be young species. So maybe we're just dealing with species that haven't had enough time to diverge and have differences fixed. So numbers 2 and 3 don't necessarily have anything to do with human error. This might actually be a quote unquote real, like biologically real and important phenomenon. Before I can go too far into this, I want to especially talk about, okay, if we're going to distinguish between my 2.1 and 2.2 up here, I need to talk a little bit about morphological differences. And for the rest of the talk, I'm not going to be thinking about morphological differences as a simple yes, no kind of thing. All right, we can have diagnostic differences between species. And so you can have a nominal species that was given one name, people looked at it better and found two different things, and you can say, okay, you know, they're diagnostically different, they're really two different species. Those things we actually still kept in our literature review because our criteria for inclusion was really like, did this thing have the same name and then get divided for whatever reason? So we, in our thinking and writing about this, have sort of been calling them pseudo-cryptic species, which is one of the names for this kind of thing in the literature. We also might have no differences between species, right? Maybe they really are exactly the same. But we have a third possibility in here, which is that there might be statistical differences between species. So maybe you don't have any single trait that's fixed between two different genetic groupings, but if you measure a lot of different stuff and look at sort of means and averages or principal components analysis, you might find some statistical differences. And here's one example of that. The Mediterranean heart urchin, Echinocardium, turns out to be 3 different groups. And if you measure any one of these given traits, there's a lot of overlap here. So you can measure any of these things on the test of the urchin that are used to call species and not come up with differences. But if you combine them, if you combine all of those traits in the principal component analysis, you suddenly recover sort of clusters of traits. And clusters of traits in a way that it might be difficult to distinguish between, you know, I have this urchin, is it a dark blue thing or a light blue thing, based on those measurements. So again, sort of to come back to these morphological differences, we have diagnostic fixed differences, which I'm again calling pseudo-cryptic species, or no differences between species. And in here, I'm lumping statistical differences for the analyses. So we do come back to a yes, no, but I want to point out that I am going to be talking a little bit about these statistical differences between groups. I've got a few more predictions to think about. If these cryptic species are only due to human error, it's all a human problem, we would expect to find more pseudo-cryptic species than things that are truly cryptic overall. We might find more pseudo-cryptic species in earlier described species, again, sort of because of technical problems. And we might find more pseudo-cryptic species, again, coming back to these biological traits, thinking about these biological traits as things that make differentiation more easy. So you might be able to like identify fixed differences as humans if they are aligned with, say, a hard skeleton. If stabilizing selection is important to come to our eco-evo hypotheses, we might expect to find more truly cryptic species in stable environments. We might expect to find more truly cryptic species in soft sediment than in rocky shore. For instance, if burrowing sort of requires an optimal phenotype, you might see selection for that phenotype. If large effective population sizes are important, we're using connectivity as a proxy for effective population size because it was easier to sort of estimate that for species than actually get population size estimates. And of course, there's our recent divergence. And I'm sorry to disappoint you. We do not currently have divergence data on the scale that we would need to actually get at this. And so that's maybe a big caveat to all the rest of the results I'm going to present, is that I'm sure this is important in some, if not all, in some cases, we know it's important, but there's also, you know, I can't present data on that today. All right, so to get at these questions, instead of comparing cryptic species to the whole worms database, I am now narrowing it down again. So instead of 977, if I'm going to talk about morphological differences, we need cases where we also have morphological measurements. And so combining cryptic species with morphological data gets us down to a data set of 492, which is smaller than we were for the first analysis, but still reasonably large. And built some generalized linear mixed models to test whether or not, test the probability that we have cryptic species sensu stricto. So these are our truly cryptic species, is what you're going to see on the y-axis. And we use taxonomy as a random variable in the models there because we needed to consider it. Thing one that we found, just sort of counting what's out there and not really doing stats on it, there are more truly cryptic species than pseudocryptic species, and that's a factor of about 2 to 1. So what we see in the literature is not, it's pretty clearly not just that there are diagnostic differences that people have identified later. We did not find any pattern associating whether or not you're truly cryptic or pseudocryptic with time. So that's sort of a mess. We also did not find any relationship between being truly cryptic or pseudocryptic and any of the biological traits. So that was sort of interesting and head scratching to us because it had been so important in the first analysis. But we're both working with a smaller data set and also asking the question a little bit differently. So we're not comparing all nominal species and say who has cryptic species or not. We're saying, okay, you have cryptic species already, what kind do you have? And so it is a slightly different question that we're getting at. Maybe more interesting results. So in stable habitats, which I'm considering deep sea, pelagic, caves, more species are truly cryptic. But from here, my co-author and I have kind of gone several rounds about how do we actually decide if something is stable or not. And really, okay, you know, how do you compare the deep sea to some of these more shallow environments, et cetera? So I'm not putting a whole lot of stock in this analysis. This is exciting to me, though. If we consider species that are in shallow water soft sediment or shallow rocky shores, so coastal rocky shores, coastal soft sediment habitats, we're down to just 193 cases. But what we see there is that if you're in the soft sediment, we have a higher percentage of those species that are truly cryptic, that do not have morphological differentiation. And on the rocky shore, that percentage is lower, and that difference is statistically significant. So this is consistent with the idea that maybe there is selection for, I'm again thinking about burrowing, but maybe that the habitat of living in soft sediment constrains morphological evolution in a different way than living on a more open rocky shore would. And there's actually some work out of fishes as well, looking at different wrasses, showing that burrowing wrasses have more constrained morphology than non-burrowing wrasses. And I was excited to find and read that paper. So does sediment constrain morphology? Not sure, but that is in line with what we've got so far. If we think about habitat connectivity and population size, that may be like neutral-ish reason that we might have cryptic species. This is our connectivity estimate. So down here, we have habitats that are super patchy. Down here is the open ocean, which we're thinking about as being very connected. What we see here is that out in the open ocean, we have a larger percentage of species that are statistically differentiated. So these are truly cryptic along with our undifferentiated, but that's where we expect to see that statistical differentiation when you have a large population size. And so phenotypes are maybe varying, but not fixing quickly in that population. So to wrap it up, and I'm looking good on time, to wrap up this set of predictions, we actually didn't find strong evidence that human error is responsible. Now, I'm not saying that it's never responsible. There are very clear cases out there where, like, yes, this is some kind of human problem. For instance, those glaucous nudibranchs that I showed you, probably related to maybe the fixative that those nudibranchs were collected in. But not always the case. If we think about stabilizing selection, we do see some evidence that maybe there are eco-evolutionary processes at work coming up with these cryptic species. And again, I can't look at divergence. So if you think about sort of what can cryptic species tell us about how evolution works in the marine environment, we see some signals that these cryptic species may actually be due to real biological processes, not just cases of human error. And again, trying to sort of situate this in the larger literature, people argue about that a lot. And there's just a lot of, I would say there's sort of strong voices on both sides. But if you look at individual cases out there, again, coming back to some burrowing worms, like the ones on the slide I just showed you, where individual research groups have actually been able to also look at divergence time. There's some evidence that, right, some of these things may be truly old lineages that are cryptic and are being maintained as cryptic through time. They're not just young species. And so something else might be going on to lead to the stasis that we see. So again, you know, to sort of summarize what I just said, we generally have support for the idea that biological factors may cause cases of cryptic species. Not always, but sometimes this certainly is possible. They're not only cases of human oversight. So to wrap the whole thing, right, here's where we are 30 years after this paper. Where are we now? We have identified nearly 1,000 cases of free-living metazoans. Again, that literature search was run through 2020, so there's certainly more described. And there's still lots out there, right? So we don't think that this is everything out there that hasn't been discovered. We have evidence that the history of science, biogeography, and maybe some biological traits all relate to whether or not you find cryptic species in a taxon. There's fewer missing chordates, or sorry, fewer missing cryptic species in chordates and in the Northern Hemisphere. So if you work on like squishy fouling organisms, you probably already know that taxonomy is challenging and it's hiding a lot of cryptic diversity. So this may not be news to you, but that is what we find in our data. And our last data set support some potential for eco-evolutionary causes of cryptic species rather than just taxonomic error. So with that, I need to thank my colleagues. And Shinwil is really the driving force behind a lot of this. Emma Shea did specifically helped us query NCBI, so that she's been really instrumental in some of the informatics. And my student, Mark Tran, who wrote the geographic code that worked, not the geographic code that didn't work. That's me. And I need to thank some funding sources, mostly from Aix-Marseille University, that allowed me to travel and continue to work on this last year. So with that, thank you very much for being here, and I can take some questions.